Okay, thank you very much for inviting me. So my talk today will be about what we called ancient DNA. It means the retrieval and analysis of the genetic information from ancient materials and fossil remains. And this is a fairly new field. It's less than 30 years old, and you can say operates on the interface between molecular biology, archaeology, paleontology, and geology. I'll start by giving you a very brief historical introduction to the field, and then I will continue with a number of case studies that my group has been involved in. So it basically all started back in 1984, where Alan Wilson's group in Berkeley showed it was possible to obtain DNA from the skin of the quagga. Quagga, it's a zebra-like animal that lived and died out in South Africa in the end of the 1800s. And then the year after, a young student at the time claimed it was possible to obtain DNA from the skin of an Egyptian mummy, several thousand years old. And then in the end of the 80s, beginning of the 90s, we had the breakthrough of the polymerase chain reaction that you can say for the first time enabled to retrieve single surviving molecules. And we saw a bust in ancient DNA claims. So you could read in the literature, it's possible to obtain DNA from things like amber and tumbled insects, dinosaur bones, magnolia leaves, all many millions of years old. However, after a few years, it turned out that this DNA sequence from the Egyptian mummy probably came from the student himself. And after a few more years, well, it turned out that all those claims we call geological ancient DNA, it means claims more than 1 million years, was in fact the result of either human contamination with amplicons, or in one case probably somebody eating a chicken sandwich in the laboratory. So quite early on it became evident that it's very easy to obtain false positive results due to contamination with modern DNA. So why is this the case? Well, the DNA molecule actually turns out to be fairly unstable, and it will decay with time if it's not repaired. And this is due to various processes, bacteria onslaught, the action of nucleases, etc. But even if it survives those processes, it will still decay due to spontaneous chemical reactions such as hydrolysis and oxidation. And the rate by which this degradation takes place is highly dependent on the environment in which you find the DNA. And I mean, many people, including myself, have tried to predict how long time can DNA survive under various settings. And in all cases, those models have proved to be too simplistic. So actually, we do not exactly know how long can DNA survive under various conditions. However, what we do know is that some environments are better than others. For example, cold environments are better for long-term DNA preservation than hot and humid environments. So how do we do it? Well, all our work today is centered around the use of next-generation high-throughput sequencing, and we use those different platforms in various ways. We're doing shotgun sequencing. We're doing in-depth sequencing of amplification products. We're doing various types of genomic scale capture that is then sequenced. What makes it difficult is that it's basically some old stuff we're dealing with, and that creates a number of problems. I mean, there's degradation products that inhibits amplification. There's various types of DNA damage, some of which result in insertion of RNA bases during amplification. But of course, one of the biggest concerns, especially in ancient human genetics, is contamination with contemporary DNA. And besides, you can say, dealing with that problem today bioinformatically, trying to authenticate that what you have is, in fact, real ancient DNA, we're also today taking various types of precautions that you didn't do back then in the 80s and beginning of the 90s. So we have complete physical separation of the laboratories where we do DNA extractions, amplification setup from the labs where we actually do sequence and cloning, etc., in order to avoid contamination or minimize contamination with amplicons. The clean labs, we have positive air pressure, nightly UV radiation of surfaces, carry full body suits, etc. And this type of, you can say, lab contamination, also it can be very annoying. It's quite easy to detect, for example, through independent replication of results by two laboratories. What is much more intriguing is what we call sample-based contamination, where the sample itself has been contaminated by people handling it, for example, which makes it more difficult to deal with. And that's the reason why modern archaeology, should be done like something like this in full body suits in order to minimize the level of contamination. Okay, so what kind of questions can we then address using ancient genetics? Well, one of the things that my group has been very interested in is extinctions. Because extinctions, you can say, is just as an important part of evolution as is speciation. And sometimes you can say you have a number of extinction happening over a very short period of time, and you talk then about, you can say, mass extinctions. And the last mass extinction on Earth is probably what we call the late Quaternary megafauna extinction. So if you go back around 12,000 years, you would see an environment in Europe, Asia, the Americas that is very different from today. And one of the things that makes it different is that at that time you had a large diversity of big-bodied mammals. So you had elephants in the form of woolly mammoth, you had rhinoceros, lions, ground sloths, big ground sloths, etc. And for unknown reasons, about 2/3 of these animals disappears presumably around the end of the last ice age. And of course, people have really been puzzled by this. What caused that extinction? And different theories has been proposed over the years. Some people believe, well, it was human that killed off all these animals, and presumably it happened very quickly in a blitzkrieg-like manner, within 1,000 years of first human contact. Others have suggested it's hyper-diseases brought to the animals by humans and the dogs. The effect was presumably the same, very quick extinction after first human contact. Still others claim, well, it was climatic changes that caused the ecosystem to collapse and caused these extinctions. And then finally, there's people claiming it was extraterrestrial impact hitting North America 12,900 years ago, creating numerous disturbances and even climatic changes. And we just actually completed, I would say, probably the most extensive study on the late Quaternary megafauna extinction to date, where we have used combined past population genetics on 6 different species, 3 that survived, 3 that got extinct, together with something called climate niche modeling and various statistical approaches on the archaeological record. And what we see is that we can definitely dismiss things like the extraterrestrial impact. I mean, just nothing happens at that time in the megafauna assembly, So, I mean, at that time, 12,900, I mean, nothing happens. We can also definitely dismiss that the extinction happened very quickly after first human contact. Humans and megafauna in many areas are coexisting for several thousands of years before extinction. And in fact, the extinction itself seems to be happening very, very slowly. We can also see that climate is a major driver of population dynamics in these animals. So it's really controlling how, the number, the effective population size of the animals changes over time. However, the climate affects the different species and sometimes even the different continental populations very, very different. And therefore, I mean, it's unlikely that a climatic event, so to speak, alone should be able to cause the extinction. At the same time, we can see in terms of human impact, in some species, in some populations, there seems to be a human effect. There seems to be a great overlap between humans and these megafauna animals. You see it in the archaeological record. In other species, they don't even overlap. And you can say the most striking thing is that there's nothing population genetically that distinguish the ones that go extinct from the ones that survives. And in fact, we also see a massive competition among some of these animals. So it seems to be, you can say, almost random events. I mean, just unfortunately events that sometimes take some of the species out while others survive. So if you ran the whole experiment again, my prediction would be that the outcome would be different. I mean, some of the guys that have survived until today would have gone extinct, and the opposite. Another thing we use ancient DNA to is to reconstruct past ecosystems. So by simply going and retrieve DNA directly from ancient sediments. And this was a discovery I did actually during my PhD, that it's in fact possible just from a few grams of ancient sediment to retrieve the DNA of the plants and the animals that lived in that region during that time. And the rationale here is, of course, The reason why we started thinking about this was that if I do like this on the table, my DNA will be on the table. The question is, how long time will it survive, right? But I remember we talked about yesterday about not listening to your supervisor. I actually remember my supervisor said, this is the most stupid idea I ever heard. But we went there and we tested it both on sediments from Siberia permafrost, which is ideal for long-term DNA preservation, but also sediments from New Zealand. And as I said, 2 grams of sediments, you could obtain DNA from the plants that live there, but not only that, also from various types of animals that are running around, taking these plants, even in the complete absence of macrofossils, of bones, hairs, anything. And of course, this was, you can say, a really important discovery in the sense that suddenly you can go into an area and reconstruct the environment, even in the absence of macrofossils that people couldn't do previously, right? And in fact, it's just being started taking up after several years by modern ecologists that are going out instead of looking at plant composition in an area, then instead they're going out taking soil samples and investigating them for DNA, right? So how can it be that you find this DNA in the sediments? Well, our studies suggest that a lot of the DNA, especially from the animals, come from feces and urine. So feces, DNA is coming out from the animal, from the intestines, and with urine from the urine bladder. And there's evidence showing that when the cells get into the sediment, they become lysed very quickly. The DNA is getting released. But because DNA is electrically charged, and many sediment particles are electrically charged too, some of this DNA will bind to these sediment particles. The final possibility, of course, is that some bacteria actually will incorporate some of this DNA into the genomes. And I will show some results just in a moment that actually where we have found evidence that this is in fact the case in some cases. You can even go a step further and you can say look at ice cores. You probably know that ice cores have been drilled all over the globe in order to investigate for climatic changes, and most of the ice is what we call clean ice. But when you hit the bedrock, you're getting what is called silty ice, full of dirt from underneath the ice cap. And then it struck me, what about could we actually use, is the DNA preserved in this dirt, so we could reconstruct how was the environment in these ice-covered regions on Earth the last time they were ice-free. And this would be significant because we're talking about 10% of Earth's terrestrial surface today is covered by thick glaciers and ice caps, and we basically have no idea what was present, during the last time they were free of ice, right? And what we basically showed was that in southern central Greenland at the Dive 3, we found evidence from a diverse conifer forest, nothing like you see in Greenland today, with spruce, pine, taxus, various types of insects. And of course, this was really exciting. But when we dated, the basal ice using various types of dating methods, you can see there's big arrow markings, but they all overlap. We could see, well, it dates to the previous interglacial, about 450,000 years ago. And this was really interesting because all the models at that time about how does the Greenland ice cap react to climate changes suggested, well, we had this global sea level rise 123,000 years ago during the last interglacial, and they believed, well, this is coming from melting of the Greenland ice sheet, the southern Greenland ice sheet during that time. That was caused this enormous sea level rise. Suddenly we could show, well, obviously some of the ice cap melted away, but it didn't disappear. It was there still. And that means there's something wrong with the models. All that water didn't come from Greenland. Another thing we have been interested in is looking at how long time can life, in form of bacteria, being preserved. There's been, I don't know if you guys have seen, there's been these claims that, spore-forming bacteria can survive in things like amber and halide for tens to hundreds of millions of years, right? But the problem with these studies is that they are, you can say, they're very easy to pick on in terms of the whole experimental setup. That it's basically caused by contamination. And we decided to investigate that using permafrost sediments and taking a very different approach. The previous studies have been built on culturing, trying to culturing, ancient bacteria. But as you probably know, only about 1% of all living bacteria today can actually be cultured, right? So you're really restricting yourself here. So instead we said, well, let's use our knowledge about how the DNA molecule is actually reacting after an organism dies or goes into dormancy. So we said, well, the longest DNA fragment that has ever been obtained from something we know is dead, namely a penguin from an ancient penguin from Antarctica, was 1 kb, 1 kilobase. So we said, if there's anything to being ancient viable bacteria, we should be able to in one go amplify 4 kilobases. That's the one thing. The next question was, we said, well, if that's possible, we want to investigate, is these bacteria in dormancy or are they actually metabolic active? Do they have any kind of DNA repair? In order to address this, we treated the extracts with an enzyme called UNG, which basically takes out, you can say, those strains that has deamination damage of cytosine, which accumulates very quickly, if an organism dies or goes into dormancy. And finally, we said, well, if there's evidence of DNA repair, there has to be metabolic activity. Otherwise, you can't be dormant. There has to be a metabolic activity. So we actually set up together with a team in Lund who had specialized in looking at tiny releases of CO2 from frozen samples, in situ. What did we find? Well, we could, in fact, amplify, under very controlled conditions, 4 kilobase fragments from the permafrost samples. When you reach around 700,000 years, we couldn't amplify anymore those long fragments. But as you can see here, the diversity, within the sample declined with time. And what does this reflect? Well, one thing it could reflect is if you have, a similar community through time, right? But you have a different death rate, so to speak, of the different bacteria types, you would see something like that. When you then look at what is the composition after the UNG treatment, what kind of bacteria survives? Well, we can see in the modern sample there's no much difference between the bacteria composition before and after UNG treatment. But when we get back to the ancient samples, you can see there's some bacteria groups that completely disappear after UNG treatment. For example, the endospore-forming bacteria like Bacillus clostridium, that we know where the spore, if they go into a spore state, have no metabolic activity, no DNA repair. They have other mechanisms that can kind of slow down the rate of degradation. But quite surprisingly, the ones that seems to survive the longest and seems to have some kind of DNA repair is in fact non-spore-forming gram-positive bacteria related to the group Actinobacter. And what is special about these guys, Actinobacter? Well, one thing that they're really good at is long-term starvation, okay? So then if you look at the CO2 release, obviously we can't signify, that it's coming from biological activity, but nevertheless it's interesting that you see the highest CO2 release in the youngest samples, and then it's basically declining with the age, and around 700,000 you can't distinguish it from the background. So what does this mean? Well, to me, I think what happens here is that you have a normal viable community in the surface layer, which we call the active layer. But when this turns into permafrost, when this goes into a frozen stage, then basically the community starts starving, right? And it dies out. It just takes, for some organisms, it just takes a long time for dying out. But the implications are really interesting, right? Because permafrost is melting all the time, completely naturally. Ancient bacteria is being released to the surroundings, right? So in principle, you can say there could be lateral gene transfer between modern-day bacteria, ancient bacteria, and even, you can say they could even compete with each other. And equally interesting, we have done a number of experiments where we have tried to basically feed bacteria with ancient damaged DNA, and see, well, can they actually take it up into the genome, right? And what we see is, yes, they can do that, and it's all kinds of damages. We're talking AP sites, multiple substitution differences, and even down to 20 base pairs, they still can take it up, right? The rate when it's these very short fragments is lower than with the long fragments, but still it happens. And of course, the implications again here are I think, very interesting, because if this is happening out in nature, we know that in the sediments, in the soil, there's a bulk, of dead bacteria DNA, and also in many cases from many different ages. And suddenly, this will, of course, be a complete new, you can say, process contributing to bacteria evolution, besides, just lateral gene transfer between, living bacteria and normal, Darwinian mutation and genetic changes. And another thing where it has, and I think it's worth considering here, is think about this biological waste, for example, gene-modified organism where you autoclave them, right? And then, it's not my area, and then they put them out into the environment. They're not finished there, right? If this is happening, then it's actually pretty significant. Okay, another thing that my group has been dealing with is early peopling of the Americas. And this is an area where, I mean, there is a massive fight going on. I mean, it's unbelievable. So you can say the general notion, I think, is that the first people coming into Americas happens around 12,500 years ago, and it's what we call Clovis. I mean, they have these special technology with big spears, etc., called Clovis points. And some people believe that Clovis, these Clovis guys, are in fact Europeans coming from Europe. There's, you can say, skull features that fits that pattern. There's also similarities in the technology. Other people believe they are ancestors to present-day Native Americans. And the first one, of course, they believe then that they were replaced, these Europeans were replaced by, later on, by Native American ancestors. Then there's still people who believe that, well, there was actually humans in the Americas prior to Clovis, okay? And I got involved in this because of this guy here, Dennis Jenkins, who is doing excavations in a set of caves called Paisley Caves in Oregon, where he finds literally hundreds of ancient feces. And we started, well, first I must admit I didn't find it very interesting to look at this, but I didn't think it would bring us much further. But it actually turned out that some of these feces contained DNA sequences that matched definitely Asians, and also with possibly, you know, ancestors to Native Americans. And when we looked at the position of these feces samples in the strata, they were actually very old, some of those, 1,000 years older than the oldest evidence of Clovis. And of course, we had two major challenges here to address. One was these feces samples, Coprolites as they're called, in fact as old as suggested from the position in stratigraphy. And that was fairly easy to deal with because it's organic material, so we sent it to two different C14 dating labs. They came back with the same result. Yes, I think it was like 4 of them was actually predating Clovis by 1,000 years. The next question, of course, could these results be due to contamination, right? Because I mean, people handling the coprolites during sampling, etc. So basically what we did, we typed, genetically mitochondrial typed, every single individual that had participated in those excavations, you know, to exclude it. And no, I mean, we could exclude that scenario. So this discovery was of course quite significant because it means that the whole idea, the whole conception of Clovis first into Americas needs revision. There was people in there before. The classical idea you see in the textbooks of people coming from Siberia, going through the interior ice-free corridor down to the lower 48 states is also wrong because the ice-free corridor wasn't open at that time. However, what is evidence that was open was a small stretch of land along the west coast, you know, during that time. So they could have come along that way. So, but I would say, I mean, when we published this in 2008, I mean, all of 2009, I was dealing with replies of angry people. So, I mean, I really hit something really bad there. But anyways, you can say that, as I mentioned in the beginning, I mean, next generation sequencing has truly revolutionized the field of ancient genetics. And it's really in two ways. I mean, both because of course, the throughput, the amount of nucleotides you can sequence in a very short time, but also, equally important, because you for the first time can target very, very short pieces of DNA, right? Because you're not dependent on primer binding sites anymore. So for the first time, you can actually go and, you know, sequence fragments that are 30, 40, 50 base pairs long. And as you can see from this figure here, the relationship between fragment length and copy numbers is kind of exponentially, right? So it really matters, you know, what is the size of the fragments. And when this second generation sequencing came up, of course, a lot of people saw the potential. However, the problem, what people really didn't realize to begin with was that, of course, when you're doing shotgun sequencing with these techniques, you're sequencing everything, right? I mean, it's not specific targets. And for example, the Neanderthal project, genome project, I mean, 99% of all the sequences coming out was microbial. And this is because bones and tooth are porous. So, you know, microbes and other contamination for that matter, also human contamination, is just penetrating very deep into the bones. So we decided, we said, we want to do the first ancient human genome, and but we have to find another solution than using bone and tooth. What about if we try to use hair instead? Because hair is also a very common part of museum collections, but hair is chitin, right? We know that hair contains much less endogenous DNA per gram of material than bone and tooth, but this is not the important thing. It's not porous. It means all the contamination must be on the surface. So we basically just took some hair, dipped it into bleach, right, and cleaned it with water and sequenced, and that's exactly how it works. And we decided then to actually do, work with a sample of the first, it's actually the only sample of the first Arctic inhabitants. Okay, so it was a hair sample that everybody had forgotten about, lying in the basement of a museum in Copenhagen for 20 years. And we said, okay, this is the sample we want to use. And in 2008, we published actually the first ancient mitochondrial genome, or even the first ancient hominid genome, mitochondrial genome. And what was interesting was that already there we could see that the mitochondria was different from what you find in Inuit populations today that is inhabiting those Arctic regions. In fact, the closest relatives was found somewhere around, you can say, close to the Aleutian Islands. So this was really exciting. And then we decided to say, okay, let's try to go for the nuclear genome. And of course, it was, at that time, a major undertaking, both because of the size of the genome compared to the mitochondria, but also because, this sample was highly degraded. We're talking about an arid sequence length of 55 base pairs, right? So, we also had to develop new software for mapping, for example. And then there was the whole issue about how to deal with contamination, how to deal with DNA damage. And you can say, in terms of contamination, we really took advantage of the knowledge that we knew that this sample had been handled massively by people of European origin, right? Especially Danish origin, but Northern European origin. And therefore, of course, we could look in the mtDNA, we could look, it was a male Y chromosome, but also look for private alleles, in the rest of the genome. And basically the contamination level is close to zero. Another thing was the damage. And there we bypassed that problem simply by just using the fusion polymerase to multiply, you can say, the amplified libraries, because we did some experiments showing that this polymerase can't bypass deamination, uracils, which is a product of deamination of cytosine, right, which is the most common miscoding lesion. And in fact, we were able to sequence this genome to an average depth of 20x coverage. Meaning that we could in fact do real genotyping on it, So we have a situation, we could map around 80% of the genome. We had this average coverage of 20x, which means that we are in a situation where we have an ancient genome which is almost as good quality, really, as a modern genome. And in comparison, you can say that the Neanderthal genome that came out, a few months later, after this one had an average coverage of 1x, right? So that's you can't do genotyping. And then we used, this genome both to try to infer something about, the appearance of the first Arctic inhabitants. We could see it's a male. We could infer what kind of blood type. Also try to infer, eye color, skin color, and such. But of course, the most important thing was that we were able to address where did these people come from? And interestingly enough, this person is not closely related to neither Inuits nor Native Americans in the New World. His closest living relatives is found in Siberia among 3 groups, one of which is found more than 2,000 kilometers away from the Bering Sea area today. So what does this mean? Well, it means that it represents a previous unknown migration from the old world to the new world that didn't leave any living descendants in the new world. So they died out, so to speak, right? And this, you can say, really shows the power of ancient genomics, because you would never, ever been able to uncover such story using modern DNA. You wouldn't see, any traces of it in the present-day new world populations. And of course, you can say it's also have a number of implications as such, that you're able to do these high coverage ancient genomes, because for the first time you can actually go and say, investigate, what genetic traits appeared in a certain population when, right? And when the prices of sequencing is dropping further, you can start asking, when do certain genetic traits actually become abundant in a population, right? With high frequency, for example, or low frequency, etc. And very recently, we have sequenced an historical ancient Australian Aborigine genome, also from a hair sample. It's about 100 years old, and the reason why we went back 100 years is that except from northern Australia, where you see a lot of admixture with Papuans, etc., all other Aboriginal Australians seems to be heavily admixed with Europeans, and this is due to policy of Australia over the past 100 years, where they're taking children away and incorporating them into white families, etc. But by going just 100 years back in time, we get a complete unadmixed Aboriginal genome, which you can say I think has implications also on the longer term, potentially medically, in the sense that if you're doing disease association studies in the future, on Aborigine populations, they are suffering from a lot of diseases with very high frequency. Well, then you would be able to have a base of saying what is actually kind of original Aborigine, what came in, so to speak, secondary. But what we use it for in our study is to address what is called, what we call the Aboriginal Australian paradox. And that is that there's evidence of modern human presence in Australia dating back 45, 50,000 years. It's actually some of the earliest evidence of modern humans outside Africa. However, genetic studies, also SNP chip data studies so far suggest that Aboriginal Australians are basically Asians. They're derived from Asians. But genomic studies show that Asians and Europeans diversified from each other no more than 20 to 30,000 years ago. Basically, the implications here is that, the first people in Australia cannot be direct ancestors of Aboriginal Australians. However, morphologically, when you look at bones and skulls, etc., actually do suggest that's the case. And here again, we see the power of this, unadmixed genome, because what we can show is, they're not Asians. What happened is that they actually diversify from the same African branch as Eurasians, but they diversify around 50 to 60,000 years ago. So much before, the split of European and Asians, and which by itself, if you hold that up to the archaeological record, actually suggests it could very well be, an independent out of Africa migration, even though it's coming from the same lineage. But what happens then is that we find evidence of a massive Asian gene flow later on, going into the population. And that's the reason when you have ascertainment bias on the SNP chip data, you actually see them becoming Asians, right? But they're not originally. Another thing we see is also of interest is that some of the Neanderthal types, there's a very significant input in the Aborigine. Another thing we are dealing with at the moment is, in fact, the origin of Clovis. As I told you, it's highly debated who was Clovis. Europeans, were they, Native Americans, what are they? And in fact, there only exists one single skeleton in the Americas found together with Clovis remains. And we have some of that skeleton in the lab, and we have completed, at this stage, the complete mitochondria genome. Now we're looking for going into the nuclear genome, but the mitochondrial genome is quite interesting. It's definitely Asian. But it's an extremely, extremely rare type, you find today. It's a haplogroup that has, even though it has been screened for specifically among Native Americans, is only found in 22 individuals so far today, of which 20 is found in South America, 2 in California. And this skeleton is up here from Montana. But I would say, we can definitely dismiss the whole idea about this European influx, right? It's an Asian, but what will be interesting, of course, from the nuclear DNA is to see, is there any ancestors of these first Clovis individuals in the Americas, or or is it an extinct migration like we saw in Greenland? Another thing which is quite, I think, interesting that we are discovering at the moment, is related to Polynesian migration. That there's this massive debate going on whether Polynesians reached the Americas before European contact or not. And there's indications of this in form of the sweet potato, the chicken, but studies so far have definitely dismissed that possibility. And complete by accident, we're screening some skeletons from an extinct tribe in Brazil, and the mitochondria is spot on Polynesian mitochondria. The question is then, because these are from the 1800s something, could that be people coming with Europeans? or is it and then just going native, or is it in fact evidence of an earlier migration? But we can address this by going into the nuclear DNA, because if you see, you can say the more Native American the nuclear DNA is, the longer back you can actually calculate, you can say, the event to have happened. We also have, quite amazingly, and I would say surprisingly to me, we have in fact success with obtaining DNA from what is called Homo heidelbergensis, which is an early hominid living in it's living in Europe. It's one of the earliest hominids in Europe. But there's only one place in Europe where there's plenty of these guys, and this is at Tapurga in Spain, in the caves there. And this is dating back 500,000, right? And it's not a particular cold environment. It's still, the temperature is stable. I mean, the average length, as far as I remember, is something like 40 base pairs, right? I mean, so it's tiny fragments. But, I mean, both the damage, this tooth we have been looking at was excavated particularly for this purpose, okay? They find that many that they can actually say, well, within a month, we'll find a new tooth for you. And the damage pattern definitely is completely agreeable with it being ancient, the fragmentation, and some of the mitochondria reads maps uniquely to chimp, I mean, which is also definitely an indication of that this is probably real. So this is ongoing work. The last project I just want to briefly mention, which I also think is pretty cool, is horse evolution, horse domestication. So we have just sequenced, not to a very high depth, but we have still sequenced the genome of a 700,000-year-old horse from Alaska, using not High-seq, but in fact we have been working together with Helicos. And this is, I mean, Helicos is not, most places not particularly popular, but I mean, they have a massive output, I mean, of sequence reads. And as you can see, it's that little is horse, right? The rest of it is just crap. So, I mean, this is one of the benefits of using Helicos in this respect. You can just bully your way through the genome, so to speak. But then we also sequenced, almost completed sequencing with much higher coverage, the genome of a 13,000-year-old horse. And then the BGI that we're working together with are sequencing the donkey genome, and at the moment we're sequencing the genomes of various modern breeds. And this, of course, is really cool because you can then start looking into, you know, what happened during horse evolution, both, you can say, natural selection over the first period, and then afterwards, artificial selection in relation to domestication. Okay, thank you very much.